Uh, in umbrella trials, specifically multiple targeted therapies for a single disease, stratified by subgroups of molecular alterations. So, in phase one, if you look at, what I understand about umbrella trials is that you could have multiple targets in a single study. For example, you could have a fed and fasting study combined. You could have a single ascending and multiple ascending dose combined. You could have multiple objectives in a trial. Dr. Market, you can.
Dr. Sinha is absolutely right. I would just like to add in the umbrella trial, particularly for phase one, is you can design one protocol in which you can achieve the effect of maybe three or four protocols. And just to give an example, that if we are doing the first in man study, that is the single ascending dose study, and there are, let's say, there are by and large, usually there are 6 to 8 cohorts, and in each cohort there are 6 to 8 subjects. And on the midway of the SAD study, when you have proved the tolerability profile of the product, one can start the MAD study at a particular level, so it can go parallel, SAD and MAD at a particular level. And with one of the dose of SAD, normally the single ascending dose study is carried out in the fasting subject. But with one of the dose, we using the same subjects in one cohort, one can do the fasting study, which is usually done, and also in that cohort, repeat the study with fed, that is after giving the food, after giving an appropriate washout period. That will give an idea about the effect of the food on the product as such, about its absorption and also the PK profile and also the safety profile. Then, so you achieved already SAD study, you have achieved what is the MAD study. You have also, when you are doing MAD study, you can also achieve the long-term tolerability, with maybe 10 days, 14 days, or even 1 month uh, for this study. Then one of the cohort one can take as the elderly subjects, and you can also get an idea about the effect in the elderly, and one cohort in which one can take only the female subjects, provided there are reproductive toxicity studies have been carried out in the preclinical test. So in SAD study, you can take the females, normal, healthy female subjects, provided the preclinical study is carried out and it is safe as so far as the reproductive system is concerned. Then you can also include one cohort with female subjects. So in one umbrella study, you can do therefore SAD study, you can do multiple ascending studies, which will also give you idea about the steady state level. You can do food effect study, you can do the age and gender study, and you can also do the subjects in the female subject study. So that is so far as the phase one study is concerned. So thank you so much.
This is typically being used by sponsors to save time and money, and where specifically predictable toxicities, when you know what is likely to happen from the non-clinical study. You are
Absolutely.
saving a lot of resources by these.
Yes, saving lot of resources, saving lot of finances, and of course the most important, the time, which otherwise would take longer time. So in one protocol, you can complete at least 4 studies.